So let's say this is thylakoid membrane. Yes. So this is thylakoid membrane. और ये जो अंदर की तरफ हम स्पेस बना रहे हैं, दिस इज थाइलकॉइड ल्यूमन. और बाहर की तरफ ये जो स्पेस यहाँ पर बना रहे हैं, यहाँ पर प्रेजेंट है फ्लूइड दैट इज स्ट्रोमा. Yes, that is a colloidal protein complex is present, which is known as stroma. So this is membrane, this is thylakoid lumen, and that outer side is representing stroma. Now in the thylakoid membrane, now there will be presence of photosystem. The photosystems, they are present. Now the first photosystem which I am forming here, that is the photosystem 2. This is photosystem 2, and you know that in the photosystem 2 the reaction center, that is P680, that is a special chlorophyll A molecule which shows the absorption maxima at 680 nanometer. Now to the photosystem 2, on the inner side of the thylakoid membrane there is attachment of one complex. On the inner side there is attachment of one complex. This complex which is attached here, that is OEC, that is oxygen evolving complex. Also known as manganese complex. इस कॉम्प्लेक्स के अंदर प्रेजेंट है मैग्नीज, 4 मैग्नीज के आयंस प्रेजेंट है. अलॉन्ग विद दैट इज प्रेजेंस ऑफ क्लोरीन एज वेल एज कैल्शियम. Then it is also known as water splitting complex. Right? These are the alternative names for the OEC. Now what happens when a photon of light, it falls on P680? When a photon of light, it is fall on P680, and when a photon of light is absorbed by P680, then there is extrusion of electrons. Yes, by absorbing the energy there is release of electron. जैसे इलेक्ट्रॉन रिलीज होगा, ये जो P680 है इसके ऊपर पॉजिटिव चार्ज आ जाएगा. Yes, it acquires a positive charge. So when it acquires a positive charge, it starts behaving like a strong oxidant. तो जब ये positive charge acquire करेगा, it will start behaving like strong oxidant. It means strong oxidant means now ये किसी और कंपाउंड को ऑक्सीडाइज करेगा, जिसकी वजह से जो इलेक्ट्रॉन्स रिलीज होंगे, that is taken up by the P680 plus means itself it will be reduced. So it will oxidize some other compound, and electrons which are released, they are accepted by P680 plus and it is converted into the reduced state. Now the question is from where it will take the electrons. Now P680 plus it will take electrons from OEC. ये OEC से इलेक्ट्रॉन लेगा. Yes, OEC, इसके अंदर present है manganese ions, and these manganese ions they can pick the electrons by splitting of the water. Water की splitting से they can pick the electrons. So अगर 2 molecules of water की splitting होगी तो this will release 4 hydrogen ions, 4 electrons and 1 oxygen molecule. So you know this very well that the one of the byproduct for the photosynthesis, that is oxygen, and where this oxygen is released. So oxygen is released in this thylakoid lumen. सो ये जो प्रोसेस है ये आपको क्लियर हो गया दैट जब भी P680 इलेक्ट्रॉन का एक्सट्रूजन करेगा तो P680 will pull electron from OEC, and in the OEC the electrons they are available due to the splitting of water. Now ये जो इलेक्ट्रॉन रिलीज हुआ है यहां पर, नाउ इट विल एंटर द इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम फ्रॉम P680, द इलेक्ट्रॉन विच इज एक्सट्रूडेड, नाउ दिस इज ट्रांसफर्ड to primary electron acceptor of photosystem 2, the primary electron of acceptor, that is pheophytin. I had discussed this in the last lecture that pheophytin is colorless chlorophyll. Now from pheophytin, electron is transferred to the quinone, which is again part of photosystem 2. That's again part of the photosystem 2, that is quinone, and from the quinone the electron is transferred to a mobile carrier. Yes, mobile carrier. This mobile carrier which is involved in the transfer of electron, that is plastoquinone. So I'm writing the plastoquinone in the abbreviated form, that is PQ. Now very important point to note down here that this plastoquinone is electron as well as hydrogen carrier. Very important point to note down here that the plastoquinone is a hydrogen carrier. अब क्वीनोन से तो यहां पर सिर्फ क्या आया है, इलेक्ट्रॉन. अब ये हाइड्रोजन आयन कहां से लेगा ना, हाइड्रोजन आयन इज पिक्ड अप फ्रॉम दिस स्पेस, दैट इज स्ट्रोमा. It will pick up the hydrogen ion from the stroma, जिसकी वजह से ये ऑक्सीडाइज स्टेट से कहाँ कन्वर्ट हो जाएगा, रिड्यूस्ड स्टेट के अंदर. So let's say this is oxidized state of plastoquinone after accepting electron and hydrogen ion. The plastoquinone from the oxidized state, it is changed into the reduced one. Right? Now from the plastoquinone, the electron is transferred to the complex, that is the cytochrome Bf complex. Yes? तो इलेक्ट्रॉन कहां पर ट्रांसफर हो जाएगा, साइटोक्रोम Bf कॉम्प्लेक्स के पास. लेट्स सी दिस वन. Cytochromes, they are iron containing proteins, so they are red in color, that's why they are known as red carriers, right? अब साइटोक्रोम BF प्लास्टोक्विनोन से इलेक्ट्रॉन ले लेगा, पर ये जो साइटोक्रोम BF है, इट कैन नॉट एक्सेप्ट हाइड्रोजन आयन, इट कैन एक्सेप्ट इलेक्ट्रॉन. पर प्लास्टोक्विनोन के पास तो हाइड्रोजन भी प्रेजेंट है, ये हाइड्रोजन आयन एक्सेप्ट नहीं कर सकता. दैट्स व्हाई द इलेक्ट्रॉन इज ट्रांसफर्ड टू द साइटोक्रोम BF, बट द हाइड्रोजन आयन इज रिलीज्ड इन दिस स्पेस. व्हाट इज दिस स्पेस, दैट इज थाइलेकॉइड ल्यूमन. तो इट मीन्स जैसे जैसे इलेक्ट्रॉन का यहां पर फ्लो हो रहा है, along with the electron transport, there is pumping of hydrogen ions, there is pumping of proton from stroma to the lumen. So we can simply say that the electron transport is doing osmotic work of pumping of proton from stroma to the lumen. Now from the cytochrome BF, now the electron is transferred to the other mobile carrier. Yes, the another mobile carrier, that is plastocyanin. I'm writing it as PC plastocyanin. They are copper containing protein, so they are blue in color. That's why they are known as blue carriers. They are copper containing proteins. Now from the plastocyanin, now the electron is transferred to the photosystem 1. You know that in the photosystem 1, the reaction center is P700. So the electron is transferred to the oxidized P700. Means P700 will absorb photon of light. Photon of light absorb karega. There is extrusion of electron. Extrusion of electron, and due to the extrusion of electron, P700, it acquires positive charge. So this oxidized P700, it can easily accept this electron. Now what will happen? ये जो electron यहाँ पर extrude हुआ है, now this electron is transferred to the primary electron acceptor of photosystem 1. The primary electron acceptor for the photosystem 1, that is FES. Yes. So you please keep this in mind that for the photosystem 2, primary electron acceptor is pheophytin, and in the photosystem 1, the primary electron acceptor, that is FES. Now from the FES, now what will happen? The electron is transferred to a carrier which is attached to the outer side of the thylakoid membrane, and this carrier is ferrodoxin. Now the electron is transferred to the ferrodoxin. Earlier view was that कि ये जो ferrodoxin है ये mobile electron carrier है. But today we know that it is not a mobile electron carrier. It is loosely attached to the outer side of the thylakoid membrane. So you can simply see here that the position of OEC and the position of ferrodoxin, that is totally opposite. This is attached to the inner side of the membrane, that is attached to the outer side of the membrane. This is working along with the photosystem 2, and that is working along with the photosystem 1. Now from the ferrodoxin, now the electron is transferred to NADP+. NADP प्लस इलेक्ट्रॉन्स लेगा, हाइड्रोजन आयन लेगा और उसके बाद कन्वर्ट हो जाएगा NADPH with the help of the NADP reductase, right? अब ये जो मैंने यहाँ पर अभी आपको इलेक्ट्रॉन फ्लो बताया कि यहां से इलेक्ट्रॉन रिलीज हुआ, it through the electron carriers it will reach photosystem 1 and then ultimately accepted by NADP plus, and it is converted into the reduced state forming NADPH. This electron transport, it is known as non-cyclic electron transport. This is non-cyclic ETS, electron transport. Why it is known as non-cyclic ETS? Electron which is released from P680, वो electron दोबारा P680 के पास नहीं आ रहा है. So this type of the electron flow, where the electron is not returned back to the P680, it is known as non-cyclic electron transport system. But in the thylakoid membrane, there is also presence of cyclic electron transport system. Thylakoid membrane में cyclic electron transport system भी present है. Let's see how this cyclic electron transport system will operate. Yes, you please see here the NADPH which is formed here. What is the fate of NADPH? Now NADPH, it is utilized for the carbon dioxide fixation, right? But sometimes what happens, let's say that the carbon dioxide fixation is absent. अगर carbon dioxide fixation absent है तो means अब NADPH is not utilized. NADPH is not utilized. So it means NADPH का amount increase होगा, right? And the amount of NADP plus will decrease. Yes. Yes, please see here. अगर NADPH use हो जाएगा, if it is used, तो दोबारा क्या बन जाता है NADP plus. रिड्यूस स्टेट से दोबारा कौन सी स्टेट में आ जाता है? ऑक्सीडाइज स्टेट में. बट नाउ व्हेन द कार्बन डाइऑक्साइड फिक्सेशन इज एब्सेंट, NADPH is not utilized, so the concentration is increasing and the concentration of NADP plus is decreasing. So what will happen? So it means now NADP plus it will become unavailable. अब ये जो फेरोडॉक्सिन है ये अपना इलेक्ट्रॉन किसको देता है? NADP प्लस को. बट नाउ द NADP प्लस इज unavailable. So when the NADP plus is unavailable, so what will happen? The electron path will change. So you see here what electron path is shown here. See here? From the ferrodoxin, now the electron is transferred to plastoquinone. And plastoquinone ferrodoxin से electron लेगा, and you know it is a hydrogen carrier. It will pick hydrogen ion from the stroma. Then from plastoquinone, oxidized state, reduced state is formed. Then plastoquinone, same thing, it will give the electron to the cytochrome BF, but the cytochrome BF cannot accept the hydrogen ion, so hydrogen ion, they are pumped in the thylakoid lumen. It means whether it is cyclic ETS or it is non-cyclic ETS, along with the electron flow, there is also flow of hydrogen ion. There is pumping of hydrogen ion. मतलब कि ये जो पंप है cyclic और non-cyclic दोनों के अंदर काम करेगा. Then from here, from the cytochrome again to the plastocyanin, from the plastocyanin to the P700. तो ये जो flow यहाँ पर हम दिखा रहे हैं, this one, this electron flow जो मैंने blue color से दिखाया है, this is cyclic ETS, right? Cyclic ETS will occur one in the condition when the carbon dioxide fixation is absent, and second it will occur in the stroma lamellae. Why in the stroma lamellae? Because just now I had discussed that in the stroma lamellae the photosystem 2 is absent. अगर photosystem 2 absent है और तो फिर कौन सा पाथवे चलेगा जिसके अंदर सिर्फ इन्वॉल्व होता है photosystem 1, that is cyclic ETS. Along with that you have to keep this in mind that in the stroma lamellae the NADP reductase is absent. तो दो चीजें एब्सेंट है, photosystem 2 and NADP reductase. That's why only the cyclic ETS will occur. And the third situation where the cyclic ETS will occur when the wavelength of light which is achieved, which is received, that is more than 680 nanometer. If the wavelength of light is more than 680 nanometer, तो ये वाला फोटो सिस्टम तो एक्टिवेट ही नहीं होगा, बिकॉज दिस फोटो सिस्टम इज एक्टिवेटेड एट 680 नैनोमीटर. सो ओनली विच फोटो सिस्टम इज एक्टिवेटेड? दिस वन. तो अगर सिर्फ ये फोटो सिस्टम एक्टिवेट होगा, ओनली वन फोटो सिस्टम, सो श्योरली विच ETS will occur, that is cyclic ETS. So these three conditions where the cyclic ETS will occur, that is very much clear to all of you. So now let's cite the important differences between the non-cyclic and cyclic one. In the non-cyclic, there is involvement of photosystem 1 as well as photosystem 2. But in cyclic, there is only involvement of photosystem 1. तो देख रहे हैं फोटोसिस्टम 1 से इलेक्ट्रॉन रिलीज हुआ, रिलीज होने के बाद इट विल फॉलो दिस पाथवे एंड अगेन रिटर्न बैक टू फोटोसिस्टम 1 P700. तो जो इलेक्ट्रॉन है जहां से रिलीज हुआ वहीं पर दोबारा वापस आ रहा है, तभी तो क्या कह रहे हैं? साइक्लिक ETS. In the non-cyclic ETS, there is formation of NADPH, but here in the cyclic, NADPH formation is absent. Now in the non-cyclic ECS, there is requirement of electron donor, external electron donor, that is water. Yes, जो नॉन साइक्लिक ETS है वहां पर जो इलेक्ट्रॉन P680 से रिलीज हो रहा है दैट इज नॉट कमिंग बैक टू दी P680 अगेन. सो दैट्स व्हाई नाउ देयर इज रिक्वायरमेंट ऑफ एक्सटर्नल इलेक्ट्रॉन डोनर. तो P680 प्लस को इलेक्ट्रॉन कौन प्रोवाइड कर रहा है? OEC, which is coming from water. So here external electron donor, that is water is required. पर यहां पर जो इलेक्ट्रॉन P700 से रिलीज होगा वो दोबारा इस इलेक्ट्रॉन पाथवे के थ्रू दोबारा वापस आ जाएगा. सो हियर एक्सटर्नल इलेक्ट्रॉन डोनर, that is not required. Now you please keep this in mind that light reaction के दो एम है, एक NADPH बनाना और दूसरा ATP फॉर्म करना. अब चाहे वो नॉन साइक्लिक ETS हो चाहे वो cyclic ETS हो, in both the ETS there will be formation of ATP, and this ATP formation which occurs in the presence of the light, you can simply call it as photophosphorylation, right? So चाहे नॉन साइक्लिक ETS हो चाहे साइक्लिक ETS हो, दोनों के अंदर फोटोफॉस्फोराइलेशन होगा. Here you can call this photosphorylation as non-cyclic photosphorylation, जिससे ATP बनेगा. यहां पर जो फोटोफॉस्फोराइलेशन होगा you can call it as cyclic photosphorylation. So these are the major differences between non-cyclic and cyclic ETS. तो ये जो cyclic और non-cyclic pathway है इलेक्ट्रॉन का, that is very much clear to all of you. अब ये जितना भी इलेक्ट्रॉन का ट्रांसफर हो रहा है that is on the basis of redox potential. Yes, this is redox potential based. The compounds which have high redox potential, they have the ability to gain electrons, right? And the compounds which have low redox potential, they have ability to lose electrons. तो ज्यादातर जो इलेक्ट्रॉन ट्रांसफर होगा वो कैसे होगा? The compounds having low redox potential to the compounds having high redox potential. Low redox potential वाले कंपाउंड इलेक्ट्रॉन लूज करेंगे और ये जो कंपाउंड्स है जिनका हाई रिडॉक्स पोटेंशियल है ये इलेक्ट्रॉन गेन करेंगे. तो ज्यादातर इलेक्ट्रॉन ट्रांसफर कैसे होगा? फ्रॉम लो टू हाई. And this type of the movement you will call it as downhill movement. But in some of the cases, the movement can also be reversed. That is, from high redox potential compound, the electron is transferred to the low redox potential compound, and this movement is known as uphill movement. तो बात समझ आ रही है? Low to high, downhill, and high to low, अपहिल. तो अब ये जो इलेक्ट्रॉन ट्रांसपोर्ट है इसमें कहां पर अपहिल है और कहां पर डाउनहिल मूवमेंट है वो यहां पर हम डिस्कस करेंगे. सो हियर आई एम राइटिंग द रफ वैल्यू, अप्रोक्सीमेट वैल्यू ऑफ द redox potential of these electron carriers. So I'm writing the rough value, approximate value of the redox potential here. Let's say for P680 the value is 0.8 and for pheophytin let's say the value is 0.6 minus 0.6. This is the value of redox potential. तो अब यहां पर आप देख रहे हैं कि जो इलेक्ट्रॉन की मूवमेंट हो रही है वो कैसे हो रही है? कंपाउंड्स हैविंग हाई रिडॉक्स पोटेंशियल टू द कंपाउंड्स हैविंग लो रिडॉक्स पोटेंशियल. तो ये जो मूवमेंट यहां पर हो रही है दैट इज अपहिल मूवमेंट, राइट? पर यहां से लेके क्वीनॉन से लेके यहां तक ये जितनी भी मूवमेंट है दिस इज अ डाउन हिल मूवमेंट, दैट इज फ्रॉम लो रिडॉक्स पोटेंशियल टू हाई रिडॉक्स पोटेंशियल. अगेन ये जो मूवमेंट है दिस इज अप हिल मूवमेंट. सो इट मीन्स कि यहां पर ये दो अप हिल मूवमेंट है और ये जो इलेक्ट्रॉन की मूवमेंट है ये डाउन हिल मूवमेंट है. अगर हम रिडॉक्स पोटेंशियल फॉर्म करके इलेक्ट्रॉन का पाथवे बताएं तो पहली मूवमेंट कैसी है, अपहिल इलेक्ट्रॉन की, फिर उसके बाद कैसी मूवमेंट है, डाउन हिल, फिर दोबारा कैसी मूवमेंट है इलेक्ट्रॉन की, uphill. So on the basis of the redox potential, if we see the pathway which is followed by the electron that resemble the letter Z, yes, that pathway resemble the letter Z, that's why it is known as Z scheme. Yes, this is known as Z scheme given by the scientist Hill. So it is also known as Hill scheme. और एक आखिरी बात और आप याद कर सकते हैं कि जो फेरोडॉक्सिन से इलेक्ट्रॉन का ट्रांसपोर्ट NADP प्लस के ऊपर हो रहा है, दैट मूवमेंट इज ऑल्सो अ डाउन हिल मूवमेंट. So this was about the non-cyclic and cyclic ETS, and till here I had explained the synthesis of NADPH. But along with the NADPH there also will be synthesis of ATP. तो अब ये ATP सिंथेसिस कैसे होगा इसको एक्सप्लेन करने के लिए आई विल बी डिस्कसिंग मिशेल्स थ्योरी. मिशेल्स थ्योरी आई विल बी डिस्कसिंग हियर, दैट इज केमी ऑस्मोसिस थ्योरी.